In this video, we are going to learn about chi-squared goodness of fit tests. Specifically, we will learn about why scientists do hypothesis testing, what the purpose of the chi-squared goodness of fit test is, and what steps we need to take to perform the chi-squared goodness of fit test. The first step is to define hypothesis testing, and hypothesis testing is used by scientists to reject or fail to reject a statistical hypothesis. And this helps us determine if the data that we collect are due to the independent variable or simply due to random chance. There are two types of hypotheses. The first is the null hypothesis, and that states that there is no relationship and no difference between two groups of data. The second type of hypothesis is called an alternative hypothesis, and this hypothesis states that observed results are due to some non-random cause. In other words, the alternative hypothesis is simply one of many possible alternative hypotheses that can be proposed to explain some phenomenon we've observed. So what is a chi-squared goodness of fit test? It is used to determine if there is a statistically significant difference or relationship between two groups of data. And it allows us to compare what we actually observed with our own eyes to what we expected to observe in an experiment. In other words, the observed outcomes are compared to expected outcomes to see if variation between them is due to random chance. And we will be using chi-squared tests to study genetic crosses. So what are the steps we need to take to perform a chi-squared goodness of fit test? The first step is we have to establish a research question. Then we have to come up with a null hypothesis and alternative hypothesis for that research question. We have to do the experiment and count observed values. And then we have to predict or determine the expected values. Then we have to plug all of those data into the chi-square formula to calculate the chi-square value. We have to determine the degrees of freedom. And we have to use the chi-squared lookup table once we select a p-value and identify the critical value to finally compare the chi-squared value to the critical value to draw conclusions. And that is the most essential part of the chi-squared goodness of fit test. So this is just a reminder that this is what the formula sheet looks like, and you will be provided with the chi-squared goodness of fit test formula and the data table on the formula sheet. So the first thing is the blue box represents the probability, also known as the p-value, and that's a 0.05 p-value. We'll be using that 0.05 p-value for this chi-squared test, which means you can ignore this bottom row of the table that represents the 0.01 p-value. The next box you see is called the chi-square, and that is the symbol we use to represent chi-square goodness of fit test. Then you see the summation symbol, and that means you should sum all of the values that come after that symbol. The O stands for observed results, and the E stands for expected results. So let's do a practice example. Our research question is, do seed color and seed shape assort independently? This is essentially assuming that in corn, the different kernels of corn and the traits that each kernel has will assort independently, and they will not be linked. The traits will not be linked to each other. So we have to use certain letters to represent certain alleles for the seed color gene, and another set of letters to represent the seed shape gene. And so we've chosen the letter capital R to represent the purple seed color and lowercase r to represent the yellow seed color. Whereas we've chosen the symbol SU to represent the smooth seed shape and lowercase su to represent wrinkled seed shape. And if we look at the corn and the different phenotypes that each kernel has on the corn, you will notice that there are some yellow wrinkled seeds, there are some yellow smooth, there are some purple wrinkled, and there are some purple smooth. So there are 4 possible phenotypes in this corn. And I will say that we are provided with this information about the genotype of the parent. So we know that the parent in this cross that we're about to do has heterozygous genotypes for both genes. They have capital R and a lowercase r, and then for the seed shape gene, they have capital SU and then lowercase su. So they're heterozygous for both genes. The second step is we have to determine the null hypothesis for our experiment. And in this experiment, the null hypothesis is that there is no relationship between seed color and seed shape. Once we have the null, now we can determine the alternative hypothesis or an alternative hypothesis. And the one I will write is there is a relationship between seed color and seed shape. I think the biggest recommendation for writing null hypotheses and alternative hypothesis is keep it simple. Null is always that there is no relationship between the two variables, and alternative is that there is a relationship. The next step is we have to count the observed values. So we have to go to the corn and actually count the different seeds of each phenotype. So let's do that. Here is the parent corn, and we know that its genotype is provided, and there are 4 possible phenotypes that we see represented in the image. So I've already counted the different phenotypes for you, and I'll provide you with the table here. So these values in the table represent the observed values for each of the 4 different phenotypes that exist. The next step is we have to determine the expected values. And this is the hardest part for most students. In order to calculate the expected values, the first thing we have to do is do a dihybrid cross for the parents, which means we have to take the parents, which are represented here in the gray box, and we have to do a 16-box dihybrid cross. So I'll represent that here, and each of these gametes that are written on the top of the box represent the gametes that are produced by one parent, and then on the side of the box, those are the gametes that are produced by the second parent. And when those 2 parents reproduce, we will produce the following offspring. And the genotypes are written in the boxes on the punnett square. The remaining offspring will look like this. And now we need to use those genotypes to determine the expected phenotype ratios for that cross. So we know, based on the genotypes that are provided, that there will be 9 purple smooth offspring. There will be 3 purple wrinkled offspring. Then there will be 3 yellow smooth offspring. And lastly, 1 yellow wrinkled offspring. So we have this 9 to 3 to 3 to 1 ratio. The next step is we have to convert the expected phenotypic ratio into expected values. And this is where it can be a little bit tricky. But what we need to do is convert the ratio, the 9:3:1 ratio, into actual numbers so that we can compare values from the observed category to values in the expected category. And in order to do that, we just need to multiply the total number of observed offspring that we counted, which is 186, by the ratio for each phenotype. So if the first phenotype appeared 9 out of 16 times, then we would multiply, and I'll show you here, 186 by 9 out of 16, which is 104.6. And then we can do that for the remaining phenotypes. So 186 times 3 over 16 is 34.9, and so forth and so on. And just to double check, we can add up all of the expected values, and we should get 186. The next step is now we have to actually calculate the chi-squared value. So we will plug the observed and expected values right into the chi-squared formula. And I've rewritten the table of those values that we just calculated here. And the chi-square formula is written here. So let's plug in the values and see what our chi-square value is. The first step is to plug in the values for the purple smooth phenotype. And you notice 118 minus 104.6. The 118 represents how many purple smooth offspring we observed, and the 104.6 represents how many we expected to observe. And we can continue to do that for each of the 4 phenotypes until we get to the last phenotype, and finally we need to add up all of the values to get one final number, and that final number represents our chi-square value. So our chi-square value for this cross is 5.52. Now we have to utilize some additional statistics tools to understand what 5.52 means. So the first thing we need to do is calculate degrees of freedom. And we are told that the degrees of freedom are equal to the number of distinct possible outcomes minus 1. So if there are 4 phenotypes that are possible, 4 minus 1 equals 3 degrees of freedom. Now we need to utilize that degree of freedom to determine the critical value and the p-value. First, we'll look at the p-value. You'll notice that we use a chi-square table to determine the critical value and the p-value. We need to find the 3 degrees of freedom column, and we need to select the 0.05 p-value. Notice there are two possible p-values that we could choose from, but we're going to use the 0.05 p-value. And we just find where those two boxes intersect. So the 3 degrees of freedom box and the 0.05 p-value, they intersect at this point. That's called the critical value, and that is shown in the red box. So our critical value for this experiment is 7.82. Now, what do we do with that? We need to compare the chi-square value that we just calculated to the critical value that was found in the table. And then we use that information to draw conclusions about the experiment we performed. If the chi-square value that we calculated is less than the critical value that's in the table, then we fail to reject the null hypothesis. Whereas if the chi-squared value that we calculated was greater than the critical value in the table, then we can reject the null hypothesis. Another way of saying this is, if the chi-square value is high, then goodbye. Now let's use this information to be able to make some conclusions. The first conclusion we can make is that the chi-squared value, which is 5.52, is less than the critical value that was in the table, which was 7.81. So if the chi-square value is less than the critical value, what can we conclude? We can conclude that we should fail to reject the null hypothesis. In other words, there is no relationship between seed shape and seed color. In other words, the traits are not linked. They assort independently of each other, just like Mendel thought. Now take a few minutes to read over this question and see if you can figure out which of the following best completes the chi-square goodness of fit test.